Hello, my name is LaWanda Hopefields, and I have the honor and the privilege of serving as the director for the Ronald E. McNair Post-Baccalaureate Achievement Program, also known as McNair Scholars. During this 2019-20 academic year, we are excited to celebrate our anniversary of 25 years on the Wichita State campus. Although during this year, we have encountered many challenges, as most of America has, we are so proud to still celebrate our students, and we have great appreciation for the faculty members who have stayed the course to bring this year's scholarly activities to a close. I would like to acknowledge my boss and mentor, Ms. Delta Q. Colvin, for her support during these most recent challenging times, but also for her confidence in me to lead this dynamic group of students. Miss Ashley Cervantes and Miss Nisha Green are the capable hands behind the scene, and a mere thank you is not enough for all that they do. They are the backbone of our program, and I am forever grateful. As we celebrate 25 years of academic excellence and 15 students who are completing their scholarly research, I also want to thank you as our supporters and as our listeners. Thank you for being there for the students and for the program as we celebrate 25 years of academic excellence. Enjoy these dynamic presentations.
So the component of my study was investigating the role of domain 4 of the anthrax protective antigen on stability and immunogenicity. And my name is Taj Allen, and it was conducted in Dr. Jim Bann's lab, a part of the Department of Chemistry here at WSU. And so the overall goal of our study is to look at domain 4 and how it fits into garnering an immune response from protective antigen, and eventually looking at that can lead to making a new vaccine for anthrax. And so the anthrax toxin PA gene, so it's a major immunogenic component of the anthrax vaccine, and the anthrax toxin neutralizing antibodies have been shown to be primarily directed at domain 4, which you can see these different colors. These are part of the epitopes of domain 4, and you can also see them a part of domain 2. And so that is another thing that the Bann lab has shown, is that mice immunized with PA alone in the absence of an adjuvant, that a strong neutralizing antibody response is generated. And so this CMG2 is a cell receptor, and it is bound to domain 4, but we also believe that it can bind to domain 2 and on this epitope right here, this red. However, if CMG2 is bound to PA and used as an immunogen, the immune response is significantly weaker. And so this graph is a toxin neutralizing antibody or total antibody response, and so are reduced when CMG2 is bound to PA. So basically, this first graph right here is showing just the overall antibody response, and then the second graph is showing the toxic antibody response, and you can see that when bound to CMG2, it elicits a high immune response, and then when bound to mutant CMG2, it shows a weakened immune response. And so you can see that the gray is significantly lower than the the yellow and red bars. And so our hypothesis is that if we can take PA and make a mutant of it, which has domain 4 removed, that we should be able to have a significantly weaker immune response, and that if the majority of the immune response to PA is because of domain 4, then we can use it for formulating a new vaccine. And so this study was carried out by using a K5 site-directed mutagenesis kit that used primers. So the primers were PAD4 deletion forward and PAD4 deletion reverse to cut the protective antigen gene that codes for domain 4 from the other domain coding genes. And so this PCR product was put through a KLOD treatment to connect the plasmid back together, so then that we can then transform it inside of E. coli to then grow, and so then we can get more of the PCR product, and then we can use that product to then test studies on. And below, you see our primers. And so this first one is the forward primer, and this second one is the reverse primer. And this is just the sequence where it was cutting. And so this is another look at it. So this is showing all of the orange is what we were trying to target and take away. And so this is just showing another visual of it. And so one of our first results was this is a gel electrophoresis. So this is showing that PCR product 1 made by Professor Bann and PCR product 2 made by me were the same DNA length. This sizes it by DNA. And so you can see on the sides that it starts from a low number to a high number. And so the farther down it gets, the longer the sequence of DNA is. And so this showed us that we got the right length of sequence, but not necessarily that we cut out domain 4. And so then we sent these off to a partnered lab, and we got these back. So these are 2 forms of DNA sequencing results. And so this red box down here is showing this is a stop codon for domain 3. And so everything after this red box is showing just the plasmid genes. So we're not seeing any sequence from domain 4, which shows that our PCR product and our whole transformation was correct. And so over here, it's showing also just the amino acids and where the stop codon, the C terminus, right here is cut off. And then the rest of this is just the plasmid DNA and showing another form of it. And so our conclusion is that we successfully made a mutant PA without D4. And so we plan to grow more of it with E. coli to determine the structure by circular diagorism, and also to look at the CD spectrum of full-length PA along with a domain 4 with only domain 4 versus the one that we have with just domains 1 through 3. And we're going to combine those together and see if that we can get the whole domain 4 spectrum, and if we'll have the same structure, have the same form, and be similar enough to show that taking out the domains doesn't do anything to the actual PA, and that if you put those 2 together, they'll still act the same. And so then we also gained information to the function and to domain 4 gives to the overall PA structure will help the understanding of the antibody epitope interaction in domain 4, and then subsequent immune reactions. And so then that will just lead to more understanding into how we can best formulate a new vaccine and then get an immune response with domain 4 missing or domain 4 there. And so here's my literature cited. And this project was supported by the McNair Scholars Program at Wichita State University, and special thanks to Dr. Jim Bann and the members of the Bann lab. So the main testing that we would have to do is take this domain 4-less PA gene and then put it into mice and then see the type of immune response we get from it, because as before I've shown, if we have a full domain, so a full PA with all domains bound to CMG2, it does elicit a immune response. And so then what we have to look at now is just if immune response will be there or if it won't. And so then if it is, then we can move on with testing to see about creating a vaccine with just the 3 domains. It was not difficult because when we first started with the gel, we saw that the different sizes, and so then that was easy to kind of see that PCR product 1 and 2 were the same length and sequence. And so then when we sent it off to the lab to be sequenced further, and when we got back was showing that, exactly where we cut it, it was a clean cut, showed that there was no any amino acids hanging on from domain 4, and that it was what we wanted at the stop codon, then starting to the rest of the plasmid DNA. So I was in Professor Bann's class, General Chemistry 2, and he was talking about it, and that kind of interested me because I'm interested in endocrinology and immunology. So dealing with the immune system and how certain viruses and bacteria attack the immune system is interesting to me. So then that's why I kind of gravitated towards this lab to work in, and just seeing the kind of protein interactions with the immune system and the antibody interactions, it's very interesting. And thank you for viewing my presentation over investigating the role of domain 4 of the anthrax protective antigen on stability and immunogenicity. And my name is Taj Allen, and again, thank you.